Hello everyone. This is the second part of lecture 7, that is enzyme inhibitors, and in this part we'll learn about irreversible inhibitors. And as you already know, there are three types of irreversible inhibitors: group specific reagents, affinity labels, and suicide inhibitors. Okay, so first let's try to get some idea about why they are irreversible inhibitors. So the irreversible inhibitors, they bind covalently with or destroy a functional group of an enzyme, okay, which is essential for the enzyme's activity, or they form a particularly stable non-covalent association. That means the irreversible inhibitors, they either bind covalently with the enzyme and destroy a functional group or a group of the enzyme which is important for their enzymatic activity, or the irreversible inhibitors, they can uh, form a stable non-covalent association with the enzyme. Okay, and formation of a covalent link between an irreversible inhibitor and an enzyme is one approach. Okay, uh, however, the irreversible inhibitors, they are a useful tool for studying reaction mechanisms, okay, such as the amino acids with key catalytic functions in the active site can sometimes be identified by determining which residue is covalently linked to an inhibitor after the enzyme is inactivated. Okay, so basically by using irreversible inhibitors, we can study which group of the active site is important for which function. Okay, now let's talk about the group different irreversible inhibitors. First, group specific reagents. Okay, so the group specific reagents react with the specific R groups of amino acids of the enzyme, okay, and thus inactivate them. And these are two example of group specific reagents, that is irreversible inhibitors. One is DIPF, diisopropyl phosphofluoridate, and iodoacetamide. Okay, so in case of DIPF, these inhibitors, it can actually inhibit different enzymes, such as it acts as an inhibitor for chymotrypsin enzyme and also acetyl choline esterase enzyme. Okay, so in case of chymotrypsin, the DIPF, it modifies one out of the 28 serine residues of the chymotrypsin enzyme. Okay, and in case of acetylcholine transferase, it's also actually interfere with the serine residue of this enzyme. Okay, and this enzyme, the acetylcholine transferase, it's important in the transmission of nerve impulses. Okay, so if DIPF PF is there, it interfere with this activity of this enzyme. So thus DIPF or the other inhibitors which can inactivate the acetylcholine esterase are uh, potent nerve gases. That means DIPF can be used as a nerve gas. Okay, so this is the photo. I mean, this figure shows how DIPF inactivate acetylcholine esterase enzyme. Okay, so say this is the part of the acetylcholine esterase enzyme, and this is the serine residue which the DIPF interact. Okay, so with this serine residue, this is the DIPF. Okay, the DIPF, it's covalently bind with this serine residue of the acetylcholine esterase and thus inactivate it. Okay. Okay, it shows another example of another group specific reagents. Okay, now then another type of irreversible inhibitors, affinity labels. Okay, so affinity labels are molecules that are structurally similar to the substrate for the enzyme. Okay, and that covalently modify active site residues. That means the substrate which are act as affinity labels, okay, they have a structural similarity with the substrate. Okay, so after binding with the enzyme, they covalently modify the active site of the enzyme. Okay, thus inactivity. Okay, so as the affinity labels, they are structurally similar with the substrate, they are more specific for the enzyme active site than the group specific reagents. Okay, in some cases uh, uh, stable molecules can be designed that resemble transition state. That means some molecules, we can design some molecules, so there are some molecules which are more specific for to bind with the transition state, not with the original form of the enzyme. Okay, and these special cases are called transition state analogs. I mean, they are also transition state analogs. They are also affinity labels type inhibitors, but they bind with the transition state. Okay. TPCK is one example of such of a affinity label inhibitor. Okay, and this inhibits the chymotrypsin enzyme. Okay, and actually it's bind with the chymotry active site of the chymotrypsin and modifies an essential histidine residue of the chymotrypsin. Okay, so this figure shows that how TPCK react with the inactivate the chymotrypsin. Okay, so this is the part of the chymotrypsin which the TPCK bind. Okay, so TPCK bind with this histidine residue of the chymotrypsin and thus inactivate, and this binding they bind covalently. Okay, this is the structure of TPCK. Okay, and this is the original natural substrate of the chymotrypsin, and as you can see, both this TPCK inhibitor and this original substrate, they have some structural similarity. Okay, so look at this. Okay, they have some structural similarity. Okay, this is another example of affinity label. Anyway, so the last type of irreversible inhibitors, suicide inhibitors. Okay, so we'll know why these are called suicide inhibitors. Anyway, so suicide inhibitors is a special class of irreversible inhibitors. Okay, and these inhibitors, I mean, why they are called suicide inhibitors? Because these compounds, that means the suicide inhibitors, are relatively unreactive until they bind to the active site of specific enzymes, and then it undergoes the first few chemical steps of the normal enzymatic reaction, but instead of being transformed into the normal product, the inhibitor is converted to a very reactive compound that combines irreversibly with the enzyme. Okay, thus and then inactivate the enzymes, and these compounds are also called mechanism based inhibitors because they hijack the normal enzyme reaction mechanism to inactivate the enzyme. So what it means, I mean, the suicide inhibitors, okay, they actually almost act as a compound for the enzyme. So first they bind, I mean, they interact with the bind with the enzyme, and the enzyme change this, actually the enzyme try to catalyze these suicide inhibitors. Okay, so they catalyze these suicide inhibitors, and in in the beginning, okay, the change of suicide inhibitors is almost like the original substrate. Okay, but the original substrate turns into product, but the suicide inhibitors, they don't turn into products. Rather, they turned into a form which is very reactive and which can irreversibly bind with the enzyme and inhibit it. That's why it is called these are called suicide inhibitors, because the enzyme is catalyzing a compound which don't become the product, rather it inhibits the enzyme itself. Okay, and this is one example, monoamine oxidase. Okay, so the NN dimethyl propargylamine. Okay, so this is one example of suicide inhibitors which inhibit this enzyme, monoamine oxidase. Okay, so let's see what this enzyme do. The monoamine oxidase, it's an important for neurotransmitter synthesis, and this enzyme requires the cofactor FAD, okay, flavin adenine dinucleotide, and these inhibitors, that is the NN dimethyl propargylamine, it inhibits monoamine oxidase by covalently modifying the flavin prostate group only after the inhibitor is first oxidized. So that means the inhibitors, okay, when it is in this form, its original form, they cannot inhibit the monoamine oxidase. Rather, okay, so look at this figure. So this is the inhibitor, okay, this pink one. So this original form cannot inhibit the enzyme. Rather, first they need to be oxidized, and this oxidation is catalyzed by the enzyme. Okay, so after oxidation, the inhibitor, they become active, and then they covalently bind with the enzyme and inactivate it. Okay, so that's why it is called mechanism based inhibition or suicide inhibition. Okay, another well known example of suicide inhibitors is penicillin. Okay, so penicillin is actually a suicide inhibitor. Okay, so what penicillin do? We know penicillin is an antibiotic, it kills bacteria, but how it kills bacteria? Okay, so penicillin inhibit the cell wall formation. Okay, so in case of cell wall, we know that the bacterial cell wall, they contain peptidoglycans layer. Okay, and peptide cross linking, I mean, when the bacterial cell wall is developed, peptide cross linking in peptidoglycans need to be formed. Okay, and penicillin inhibit with this event. Okay, they inhibit the peptide cross linking, thus they inhibit the cell wall formations. So as a result, the bacteria die. Okay, so now how penicillin do that? Okay, okay, so there is an enzyme transpeptidase. Okay, so actually this transpeptidase enzyme, it synthesizes the peptide cross-linking formation. Okay, and penicillin inhibit this enzyme. Okay, by a mechanism based or a suicide inhibition mechanism. So how they do that? Okay, look at this photo. So this is the structure of the penicillin. Okay, and the penicillin is a beta lactam inhibitor, so they have beta lactam ring in their structure. Okay, so this original form, they actually cannot inhibit the enzyme. Okay, rather I mean, when this inhibitor, they interact with the enzyme, first they get activated. Okay, so actually the enzyme, they, I mean, when, okay, so if I mean, this is the penicillin. So to act, this first this the beta lactam ring of this penicillin need to be open. Okay, so this is done by the enzyme itself. Okay, so the enzyme, that is the transcriptase enzyme, it catalyzes or activate the penicillin by opening the beta lactam ring. So once this structure is open, then it become reactive, then it can inhibit the enzyme. Okay, so that's why it is called, I mean, that's why penicillin is a suicide inhibitor, because the original structure of the penicillin cannot actually inhibit the enzyme. Rather, first they need to get activated, which is done by the enzyme itself. Then the activated form of the penicillin can inhibit the enzyme. Okay, okay, so that's all about the enzyme inhibitors. Thank you for your patience.